Hello everyone, and welcome to this podcast for cell biology. In this podcast, we're going to talk about enzyme-coupled receptors. And we're going to focus almost entirely on receptor tyrosine kinases, which we'll abbreviate as RTK. And the reason we're going to focus on the RTKs is because they are the largest class of these enzyme-coupled receptors. Over the next several minutes, we're going to talk about some general features of tyrosine kinases, receptor tyrosine kinases, and how they are activated. Then we're going to spend some time talking about two specific examples, and that is how RTKs can activate Wrasse. And then we'll spend some time talking about the pathway involving phosphoninositide 3 kinase. And we abbreviate that as PI-3 kinase. And then, I don't know if we'll spend much time on this, but I'm going to talk a little bit about Notch. So let's go ahead and get started with the general description of RTKs. So activation of RTKs, these receptor tyrosine kinases. So let's begin this discussion by first drawing our membrane. And let's draw our two inactive RTKs. So inactive receptor tyrosine kinases. Now, the reason they're inactive is because they're not associated with each other. To activate them, they need to come in contact with each other. And the reason that they have to interact with each other is partly due to the fact that they are a single pass alpha helix. Nothing wrong with that. It's just that upon binding any given signal, having a substantial conformational change, like we see with the G protein coupled receptors, having that conformational change that leads to an activity is less likely. And they also need to come together because each of them, in most cases, so each will phosphorylate the other. So let's point out some structures here. This top part, this is the cytoplasmic signal binding domain. Whereas this part here is the kinase domain. So what happens is typically we'll have a signaling molecule, and in this case we're going to draw it as a dimer that binds to each other like so. And when it comes in here and binds to one half of the RTK, the other half of the signaling molecule will bind to the other, and it will bring these two RTKs together. So let's draw it down like so. And so now this will come along here and bind to these two binding domains. And since they're connected here, they're going to bring these two RTKs next to each other, allowing the kinase domain, and remember kinases are the enzyme component that phosphorylates a substrate. So these kinase domains here will phosphorylate the other RTK. And now this will be an active RTK. And though I just drew one phosphate group here, depending on which one we're talking about, it can have numerous phosphates added to it. Now, once it's phosphorylated, they will bind a whole series of intracellular signals. There may be as many as 10 to 20 different proteins or targets that will bind to the active RTK. And now these that just bound to the RTK are active. I'm only drawing these few ones here, but remember we can have many that are binding here. Upon activating these separate proteins here, downstream intracellular signaling occurs. These different ones that are bound here, they may activate different pathways. They may activate pathways that are coordinating with each other. It just depends on the RTK that is being activated. So these are some of the key features of activating an RTK. The inactive exists as these two separated monomers that have these kinase domains that once they're brought together by a signaling molecule, activates these kinase activities so that they can phosphorylate each other. Upon phosphorylating each other, they will recruit various targeted signaling proteins that then will go on and activate downstream intracellular pathways to respond to various environmental cues that are brought in by this initial signaling molecule. Now what I would like to do is discuss one of these pathways in a little bit more detail, and that pathway is involving Wrasse. So let's talk about Wrasse. And the reason we want to talk about Wrasse is because that most RTKs can activate Wrasse. Wrasse is a small GTP binding protein that is bound to the plasma membrane by a lipid tail. They play many cellular roles. I'll mention two here. One is mediating cell division, and another is the development of vertebrate neurons. Wrasse functions as a molecular switch, just like we've talked about before. So remember, like all GTP switches, when it's bound to GTP, it's active. When it's hydrolyzed to GTP and it's bound to GDP, it's inactive. And we're going to talk about in a moment how we go from an inactive to an active, or from an active to an inactive, in just one moment. But in talking about it, we're going to talk about these two proteins. One's called a GEF, G-E-F, and this is important from going from a GDP to a GTP. GTP exchange factor. And then the opposite of that is GAP, which hydrolyzes GDP to make GDP. When it's active, it will activate a chain serine or threonine kinases. So let's draw this out. So here's our membrane, and let's go ahead and draw our already activated receptor tyrosine kinase. So they will have several phosphates on them, and we should draw the signaling molecule up here. Once activated, they will first bind to a adapter protein, which will then bind to this Wrasse GEF. That's going to exchange the GDP for a GTP. So up here, let's draw our inactive Wrasse. And it's inactive because it's bound with GDP. So this Wrasse GEF will activate inactive Wrasse by changing GDP for GTP. Once GTP is bound, it is now active. The active Wrasse now will activate this protein here that is called MAP kinase, kinase, kinase. That's 3 kinases. This being a kinase, it's going to phosphorylate its target protein. And this will be the first one that's in the cytosol. Up to this point, they've all been attached to the membrane. And so it will add a phosphate group. And so it will add a phosphate group here to this target protein, which is called a MAP kinase kinase. And now it is active. Prior to the phosphorylation of it, it was inactive. Now this MAP kinase kinase will phosphorylate its target protein and make it active. And the name of its target protein will probably not be a huge surprise. It's called MAP kinase. So we go from 3 kinases to 2 to 1. These are present in high concentrations, and they can be activated by various signals. Because of this abundance and their important role in cellular activity, they can have several downstream targets. So what does this MAP kinase do? Well, it could do several things, but ultimately it's going to have to act on yet another protein to phosphorylate it. And together they're going to change the overall gene expression. They may do so by changing a protein activity directly, or they may change expression of a gene by entering the nucleus. Now, what do these changes in protein activity or gene expression do? Well, they can do several things, but one thing they do is cause cell proliferation. And because of that, Wrasse here is considered a oncogene. We haven't talked about cancer yet, but for now what you should know is that Oncogenes are genes when they're overexpressed can lead to cancer. In this case, with Wrasse being overexpressed, that is unable to be inactivated, it will allow this entire pathway to proceed without the ability to control it. And these proteins that are being changed, or the expression of genes that are being changed, that drive cell proliferation, can lead to cancer, because one of the hallmarks of cancer is increased and uncontrolled cell division. Okay, that's all I have to say about Wrasse, but now let's talk about this second pathway. Okay, now let's talk about receptor tyrosine kinases and how they activate. So next let's talk about the pathway involving phosphoinositide 3 kinase, PI3 kinase for short. Activating PI3 kinase is linked to cell survival. And I'll explain that in a moment, but let's first look at the pathway here. So shockingly, I'm going to draw a membrane here, and then I'm going to draw an activated tyrosine kinase. Again, it's active because it's bound these signals to bring the two monomer proteins together, to dimerize them. And now it allows them to become phosphorylated and to make this RTK active. And instead of just calling this a signal, let's indicate it as a survival signal. Once this RTK here, and we might as well label it so we don't forget, is active, it will bind this cytosolic protein called PI3 kinase, the one we're talking about here. And it binds specifically to this phosphorylated RTK. And so now PI3 kinase is active. When it is active, it will bind to this molecule that's stuck in the membrane called inositol phospholipid. It already has a couple phosphates on it, but the PI3 kinase will now add additional phosphate to it. So now it's a phosphorylated inositol phospholipid. So this inositol phospholipid now recruits two kinase proteins. The first one I'm just going to call, as your book calls it, a protein kinase 1. This other one is a pretty famous kinase, and it's called AKT kinase. Protein kinase 1 will add a phosphate group to AKT, and in addition there is a cytosolic kinase that we'll call protein kinase 2 that will also add a phosphate group to AKT. AKT is now active. So when it's phosphorylated by these two kinases, it becomes active. So let's talk a little bit about what AKT does. Okay, let's focus now on what AKT is doing to promote survival. So we can draw AKT here, and then I need to introduce two new proteins for you. One is called BAD, and the other is called BCL2. And so this complex, this BAD BCL2 complex, as drawn when they're bound together, is inactive. BCL2, I should tell you what its function is. It's an apoptosis inhibitory protein. So if we could activate it, it would block apoptosis, or what we just might call cell death. But as long as it's bound by BAD, it cannot inactivate cell death, or inhibit cell death, I should say. So what AKT can do is it will phosphorylate the BAD protein. And when it does that, it changes its conformation in such a way that it can no longer bind to BCL2. And so that releases BCL2, which is now active, to go down and activate other proteins to inhibit cell death. So AKT, which is activated by the PI3 kinase, can promote cell survival by inhibiting cell death. And it does so by phosphorylating BAD, which releases BCL2, which is the protein that actually goes on to inhibit cell death. Now, AKT can also promote survival in another way, but it's still activated in the same pathway. So activated AKT can activate a protein called TOR, T-O-R. So when TOR is activated by being phosphorylated by AKT, TOR will activate several pathways that lead to inhibition of protein degradation, and another pathway involved in stimulating protein synthesis. And so by inhibiting specific protein degradation and activating specific protein synthesis, this leads to cell growth. So, activated AKT inhibits apoptosis or cell death and activates cell growth. So kind of hits the survival at two different levels, preventing death and promoting growth. Now the last pathway I want to talk about is a pathway involving this protein called Notch. Notch is an example of a receptor that can quickly move a protein to the nucleus. So let me draw how this works here. So we'll draw one cell here and then another cell here. This cell will have a signaling molecule attached to it. It will bind directly to this Notch receptor. And again, remember this was the signal on this cell. Now, the Notch receptor has an interesting domain here that, when activated by this signal here, will be cleaved off, releasing this terminal part of the protein. This terminal part of the protein is a transcription regulator. It will now move directly into the nucleus. In the nucleus, it will bind to Notch responsive genes. So it will induce the transcription of a very specific class of genes that only respond to Notch. And when it binds here, it will make the messenger RNA. The messenger RNA will leave, and it will make the Notch-specific protein. I should have also pointed out that Notch is not an RTK kinase. It's a separate one. I should have mentioned that in the beginning, but just so that's clear. Okay, that's all I have to say about enzyme-coupled receptors. If you have any questions at all, please make sure you come see me. If not, I'll see you in class. Bye.